Hi guys, how are we doing today? Welcome back to GaySMR. GaySMR, GaySMR, GaySMR. So I tried moving the microphone and the computer around a little bit to give me a little more room to work. Maybe this will work out better. My first video, you couldn't really hear it, like at all. So, we're gonna try new things today. Alright, guys. Excuse the voice crack. I have a little bit of a hoarse throat. It's very cold outside. So, today, I will be reading part of one of my articles for class, and it is called RNA virus spillover from managed honeybees, otherwise known as Apis mellifera. Apis mellifera, Apis mellifera, Apis mellifera, ASMR. And that RNA virus spillover will be to wild bumblebees, otherwise known as Bombus, the genus Bombus, 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 Bombus. Alright, and I will be moving in between softly spoken and whispers, soft spoken to whispers. I'm actually going to move back a little so the soft spoken isn't too harsh. But before we get started, I am going to light an incense. Forgive the candle that I burned down here. Alright. Oh, I love that sound. Alright. There we go. Get some frankincense in the air. Alright, we're gonna fiddle with this a little more just because the triggers are good. Oh, I wonder. Oh, can you hear the butane? Oh, I loved that. That was cute. I liked that a lot. Oh, burned my finger a little bit. Before we get started reading the paper, I'm only going to read about half of it too, because I feel like it's going to take a while to explain everything out. And I don't know if my computer can handle that much room, if I'm going to be real. So, we're going to interlude with a little bit of something. We're going to put on just a little bit of eyeshadow just for you guys, alright? This is MAC Blue-Brown Pigment. And it is the easiest motherfucking thing ever. I'm going to grab my palette that has a mirror. Our Venus palette. I don't know if I can do the pose that she's doing. I'm going to try though. I love the Venus palettes, but I sure as fuck love the MAC Blue-Brown Pigment. That shit is on point. I'm going to throw a special shout out to Casey. Thank you, Casey. This is still your, this is yours. You gave it to me oh my god, I want to say it was holy shit, was it 4 years ago? I don't know if this is iSafe anymore. I really don't care. Alright. So there is a lot. I'm a little scared to tilt it over, make sure. There's oh crap, a bunch just fell out. there's so much pigment in here, you will never run out. Like, I don't care if you wear this every day, it'll last you 4 years. I don't wear it every day, so it's like still pretty full. So I'm gonna go ahead and use what fell out on my hand. Alright. It's such a beautiful shift. This like peacocky blue. Then it shifts to a brown red. Let me blend it out for you guys. Oh my god. Oh my god. Oh my god. Can you hear that? I'm blending it out too much. It's gonna be all brown. All brown. That pigment is wonderful. 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 Wonderful pigment. Oh, I got a little bit of a tan now. Oh, you can't see it on camera. Never mind. Alright. This stuff is just so easy. Oh, I got some on my laptop. Let me blow it away. Oh, I love it. Alright. Yep, you can see my eye good. It's so easy. Literally, I just smack that shit right the fuck on. I stamp where I want it to be really, really heavy. So like a lot of the blue shows. I might need to grab some more to get down to the lash line because I'm fucking doing liner right now. Fuck that bullshit. Alright, as you can see, the lovely blue. Then I'm just going to take a little fluffier brush and just kind of just to blend out. Blend out these corners. You get a lovely, oh, my eyes are too hooded for that. Oh, I love how that looks. It just looks so pretty. I wonder if I can, just trying to take some of the excess, just whatever's left on the brush, and dragging it under the eyes. Just give it a little more pigment. Might have to go back in. Oh, so fucking pretty. Oh, I love that. Oh, I love that. Oh, that's pretty. Let me get in close. Is that patches? Are you fucking kidding me? Oh, mother. This is why you look in multiple mirrors, or at least multiple viewing lenses. Oh, I didn't put primer on either, so you know, fuck that. Oh, I love it. Love, 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 love. I'm gonna go ahead and do that other eye real fast. Maybe throw in a couple of triggers. Maybe I'll apply some to you with a little. Oh, I get the mic brushing. It's very fun to do. Oh, God. That's beautiful. I'm going to turn this way. Maybe you can see me apply it to the eye a little better this way. I always do it in a very, like, rhythmic motion. I always line my eye with the shape that I want with a very flat brush like this. Get a little bit of the pigment on and just right where it needs to go. Oh, Lord. I got scared because it looked like there was going to be a lot of fallout for a second. Just straight blue. If you don't blend it out, you get this straight up blue. Just the other side. And then when you shift in the light, it looks brown. Oh, Mac done did it. That's all I gotta say. Oh, fuck me up. Oh, can you hear it? Oh, you can. Alright, let me grab some of that. I want to disperse it into the brush. Flat brushes, for anyone who doesn't know, I mean, I'm sure if I'm talking and you guys are liking the makeup, you know what I'm talking about. But for anyone who doesn't, flat brushes are really good for just stamping pigment on at a really good, precise location. So that way you don't have to use your fingers and turn your hand into this. Oh, it's so pretty though. I just want to like bathe in this. Oh, that'd be cool. I want to do that. Alright, and then I'm actually going to swivel this around in here. Grab just a little bit of the pigment on the top of the jar. Just to help blend it out a little bit. Because I don't want to go into the actual jar with this because it's going to do a lot. Alright, now let's flick up a little bit. Take it down here. I don't think these eye shapes are even. That is alright. This is just GaySMR. Oh, we're online, bitch. Fuck it up. I'm gonna take a little bit of aura. I think that'll be a good one. This one right here. I'm gonna take a little aura and just pop it in the inner corners just for a little extra highlight. Was that enough? That's enough. I'm gonna turn this way for. Oh, fuck. Can you see that on camera? A little bit. Can you see that on camera? Oh, what the fuck? I was looking at the wrong eye, bitch. Oof. We're going heavy inner corner right now, and I ain't mad. How's that look? Oh my fucking god. That looks like how I wanted it to. Exactly how I thought it would. I love that. Oh. I love lid sounds. And the fun thing about these pigment jars is, can you hear it lock in place? Oh, just so it never spills. It's beautiful. I'm gonna put this away real fast. I'm gonna go ahead and stamp. Oh my god, I got it all over the camera. Wonderful. All onto your eyes. Now we can blend it out. Blend it out. I'm gonna use this fluffier brush for you guys. Just to do. Just to get a little going, you know what I'm saying? I'm gonna put the rest of these brushes away because I got a shit ton out. I don't know why, it's literally one fucking product. Now that we've got our incense burning, everything good, take a hit and then talk about Apis mellifera an RNA virus spillover. Oh, someone's car out there sounds disgusting. A little bit of metal on metal tapping. Alright, let's get this party started. Alright. Today we will be talking a little bit about RNA virus spillover from managed honeybee populations, Apis mellifera, to wild bumblebees, the Bombus genus, and then anything under that, whatever the species name is. So let's go ahead and talk about the abstract. Let me get my red pen out and get some good notes going on. The decline of many bumblebee species has been linked to an increased prevalence of pathogens caused by spillover from managed bees. Okay, so they know they're viruses. Wonderful. Although poorly understood, RNA viruses are suspected of moving from managed honeybees to wild bumblebees through shared floral resources. Okay, so I'm getting from this that there are viruses, and they're somehow being transmitted when a honeybee lands on a flower, and then a bumblebee goes to the same flower. We examine if RNA virus spillover from managed honeybees. We examined. Oh my God, can I read? We examined if RNA virus spillover from managed honeybees, the extent to which viruses are replicating within bumblebees, and the role of flower transmission. Okay. So I'm just going to underline that because even though that is a very interestingly worded sentence, it's the point of the paper. They want to see if viruses are going from honeybees to bumblebees. Prevalence and active infections of deformed wing virus, DWV. Deformed wing virus. That's an interesting virus. What the fuck does it do? Were higher in bumblebees collected near apiaries when neighboring honeybees had high infection levels. Okay, so yeah, honeybees are sick. The bees around them are getting sick. Oh, we found no DWV. What was that called again? Deformed wing virus in bumblebees where honeybee foragers and honeybee apiaries were absent. The prevalence of black queen cell virus. What the hell does that do? Oh my God, that sounds bad. Was also higher near bumblebees collected near apiaries. Furthermore, we detected viruses on 19% of flowers, all of which were collected within apiaries. Our results corroborate the hypothesis that viruses are spilling over from managed honeybees to wild bumblebees, and that flowers may be an important route for transmission. I don't know if 19% sounds like a lot to you guys, but if 19% of flowers near apiaries have a virus on them, that's kind of a lot, because the virus can, like that 19% of flowers can infect multiple bees. Any bee that lands on that has a chance of getting infected. So that's not ideal. I don't know what these viruses do, but they're probably not ideal for anything going on. Alright, so now into the introduction. Oh, you know what? I should probably underline some of this. We found no DWV in bumblebees where honeybee foragers and honeybee apiaries were absent. Black queen cell virus was also higher in bumblebees near apiaries, and we detected viruses on 19% of flowers within apiaries. Alrighty, let me go ahead and grab some more pens out. Pens. Alright, blue, green, and purple. Do I need orange or yellow? It's a yellowish start this. Many infectious diseases are caused by generalist pathogens that infect multiple host species. For pathogens capable of infecting multiple hosts, spillover occurs when the pathogen is introduced and transmitted from a reservoir population into a native host population. Pathogen spillover between managed and wild animals causes species to decline and threatens global biodiversity and alters ecosystem function and services. That's basically saying that this is one of the big reasons why there's such a, I don't want to call it an embargo, that's not the right word. Traffic of like different species in and out of areas is so highly regulated because diseases can be transmitted from the species you're trying to bring in. So like if you're trying to bring cattle in, there's a possibility they have a virus and then it'll like screw over the native populations. That's not something you want going on. It really lowers species diversity, and that can have a big impact over a long range of things. Like if you bring bees in to help pollinate the area, but they kill the rest of the native bees, pollination can go down, then fruit production can go down, food production can go down. It's just not ideal. Given recent declines in managed honeybees, the importance of native pollination has risen to global attention. Many of the threats to managed honeybees are also affecting native bees. Most notably, the increased prevalence of pathogen putatively caused by spillover events from managed bees. Although poorly understood, RNA viruses are suspected of moving between managed honeybees to other insects, including wild bees. Once considered to be specific to European honeybees, RNA viruses have now been... Oh, good sounds, good sounds. I don't know if any of you guys like paper rustling sounds, but if you do, I'm glad. Oh shit. Once considered to be specific to European honeybees, RNA viruses have now been detected in a wide range of insects, including bumblebees, solitary bees, hoverflies, wasps, and ants. That might sound like a lot of different things. Those are all in the same family of Hymenoptera, which is social insects. So, it's a lot, but also at the same time, it's not jumping from like a bee to a mantis or a bee to a ladybug. It's not ideal though. Due to their high mutation rates and short generation time, RNA viruses are likely to cross species barriers and adapt rapidly to new host environments. Both relatedness and shared foraging habits may increase risk of disease transfer among managed honeybees and wild honeybees. In the UK, sympatric bumblebees and honeybees are infected by the same deformed wing virus strain, and virus prevalence in honeybees is a significant predictor of virus prevalence in bumblebees. Bumblebees. Bombus. Let me get some chapstick on and then we'll do some sounds. Bumblebees. Yeah. I love putting chapstick on. It's like one of my favorite goddamn things. Oh, that was loud. Sorry, guys. Alright. Although there is some evidence of virus spillover from managed honeybees into wild bees, more work is needed to educate transmission routes. The principal directionality of virus transmission and whether once contacted, viruses replicate in bumblebee hosts. That is something to consider because sometimes viruses can move to a different host. It's called a vector species. And even though they're in the vector species, like, what's a good example? I think swine flu, this is a good one. So swine flu reproduces readily in, pigs, but it doesn't reproduce too well in birds. However, they are both vectors for the virus for each other. That's why every once in a while we get a really, really, really bad flu, because either a bird will shit on a pig and the pig will eat it and then it'll get into the intestinal tract, and they're close enough that it's like, oh, well, carry it. But then once it gets, like, it'll get processed and turned into food, and then once we come into contact with it, it gets us really sick, like 2010 swine flu level sick. Not ideal. I don't know exactly how accurate that was. I know they're a reservoir species, but don't quote that one to everyone. The general theme is right, but I think I might have gotten my exact science wrong. Horizontal transmission routes for viruses among bee species are currently suspected, but largely unconfirmed. Horizontal transfer just means moving from one species to another. So, like, pigs to birds, pigs to humans, humans to birds. Flies to humans. That's actually a big one. African sleeping sickness. Transmission may occur through the use of shared floral resources, either by contact with the feces and glandular secretions of worker honeybees or the pollen loads. However, to our knowledge, only one study examined bee viruses in pollen collected from flowers, and this was done, in a single honeybee apiary. That's not ideal. More work definitely needs to be done. We need to see kind of like a more holistic view of everything. Holistic. Oh, I love how my eyes look. Just holistically, I look like a Venus peacock. Like, oh, I would be Hera's favorite fucking human right now. Hera, like the queen of the goddesses, she loved peacocks. Beyond the point. Thus, the degree to which viruses can be horizontally transmitted within flowers acting as a bridge remains poorly understood. Fair enough. Here we assess the evidence for the spillover of RNA viruses from managed honeybees into wild bumblebees, and if transmission is facilitated by the shared use of floral resources. First, we examine the presence of RNA viruses and whether the viruses were actively replicating into bumblebee species. Quick history on viruses. It's been a semester or two since I took virology, so I'm a little rusty, but if there are three things that a virus or a cell can be to a virus, it can be permissive. I want to say it's habitable, but it's another word. Oh, shit. Permissive means that the virus can get into the cells. The habitable, I'm calling it habitable, there's another word for it, just means that it is a good environment for the virus to grow. And then the other one is just, it can't get in. If a virus, or if a cell is permissive but not habitable, the virus can enter, but it can't grow. There are cells that can be habitable to viruses, but the virus just can't get in, so it can't get infected. So they're trying to see if the virus that is in the honeybees can get into the cells of the bumblebee, and not only get into the cells, but actively reproduce, causing more viruses, causing them to also be infectious. I just love this. I love seeing this. Just that. Okay. We then examined if virus prevalence, load, and active infection in bumblebees correlated with the presence of managed honeybee colonies and their virus loads. How much virus is in one individual in a species compared to the other. We also investigated transmission through shared floral resources by testing flowers connected from our field sites and examining how bee abundance, honeybee colony presence, and landscape level floral density. That's a really, really long drawn out way of saying how many flowers are in an area. Influence patterns of virus prevalence. Okay. So given this, the researchers are trying to test to see if the amount of bees is impacting how much the virus spreads to native bumblebee populations. The amount of flowers is important because if the flowers are carrying the infection, then the density of the vector, which in this case would be the flower, is highly correlated to how many bees could be infected should the virus be able to affect the bees. And currently it looks like it is, so that's not ideal. They want to just see how it's working. I'm going to skip the materials and methods, field sampling. I kind of think I want to skip over virus detection and quantification. I can't believe I caught this with my pinky. Holy shit. Negative strand detection, sequencing, and trying to figure out what the virus is, data reporting, data processing analysis, statistics. Guys, if I'm going to be real, I'm going to skip over all that. I don't think it's it's terribly important, but I don't know how many of you guys are going to go out and reproduce this experiment with. Let's look. Yeah, I don't know how many of you guys are going to go out here and reproduce these with Quigan Arnese mini kit protocols. Like, it's not really exactly feasible for you guys. I don't think it really truly applies. I'll definitely give you guys the link to the paper, though, in case anyone is, like, extra curious. Extra curious, extra curious, extra curious. So let's go into the results. We detected black queen cell virus in, holy shit, 75.7, and deformed wing virus in 9.3. Okay, very different numbers of bumblebees tested. We did not detect Israeli acute paralysis in any of the bees. Well, that's good. And the virus prevalence in honeybees was 100% for black queen cell virus and deformed wing virus. And, okay, the amount of viruses in each cell of honeybees, and 100% of them are infected, for black queen cell virus was 6 to the 10th to 6 to the 9th. Or 10 to the 6th to 10 to the 9th. That's a lot of zeros. To the 9th, just, that's how many zeros are on this. Like, holy shit, that's a lot. And 10 to the 4th to 10 to the 10th for deformed wing virus. Honeybee deformed wing virus loads followed up bimodal distribution. S1 figure. I don't know what that means. You know what? I don't have the S1 figure. We'll get on with it. With clear separation between two groups, which we designated as either having a low, under 10 to the 7th genome copies, or high, above 10 to the 7th genome copies of the virus load. The prevalence of black queen wing virus was significantly higher in Bombus Pimaculatus. 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 At 86.3% compared to Bombus vaginus. V-A-G-A-N-S. Vaginus. And they only had 65.9% infection rates. It's lower, but it's still fucking high. Jesus. But deformed wing virus was similar in both bumblebee species. Oh, okay, cool. didn't give me the number on that, so lovely. Bumblebees collected within 1 kilometer of a honeybee apiary had a significantly higher prevalence of black queen cell virus and deformed wing virus compared to bumblebees collected from sites without an apiary nearby. Bumblebee virus prevalence was highest at sites with high honeybee abundance. In sites without a commercial apiary and with no honeybee foragers observed during collection, 37.5% of bumblebees were positive for black queen cell virus, and none, absolutely none, had deformed wing virus. Virus load was not significantly affected by apiary presence for either virus. So that just kind of means if an individual was infected with a virus and it was not near an apiary, it had the same amount of virus in its cells as one that was collected near an apiary that was also infected with the same virus. So the distance from the apiary doesn't really affect how much virus is in the organism. Once they have the virus, and it reproduces to these levels. Alright, the prevalence of deformed wing virus in bumblebees was significantly higher in sites with high honeybee deformed wing virus loads compared to bumblebees collected from sites where deformed wing virus loads in honeybees was low. Okay, I mean, if there isn't a lot of virus in the area, it's harder to get. But if there's a lot of virus in the area, it's easy to get. Virus loads. Oh, thank you for telling me this now. Virus genome copies per bee. So it's not per cell, it's per bee. So an individual. In bumblebees range from 10 to the 4th to 10 to the 7th for deformed wing virus and 10 to the 4th to 10 to the 8th for black queen cell virus. Bombus bimaculatus had significantly higher viral loads than Bombus vaginus for black queen cell virus, but not deformed wing virus. Okay. By amplifying the negative strand, that's a lot of virus talk, and the negative strand is something that I don't think I want to get into. Ask me down in the comments below if you want to know anything more about like the different genome types of viruses. We detected actively replicating black queen cell virus and deformed wing virus in both bumblebee species. We found black queen cell virus replication in 17.2%, deformed wing virus replication in 6.3% of bumblebees. Actively replicating black queen cell virus infections were higher in Bombus bimaculatus compared to Bombus vaginus. Deformed wing virus was actively replicating in 7.4% of Bombus bimaculatus species and 5.2% of Bombus vaginus. Okay. The deformed wing virus might have a very different transmission route or significantly different pathogenesis because it's not replicating very, very, very differently. Whereas black queen cell virus is, that's got a difference in species. Like, that's really hidden bimaculatus hard. Replicating deformed wing virus was more prevalent in bumblebees collected near honey apiaries. However, this is another. Okay, we've already been over this. Oh, this is the figure. I'm gonna hold this up. This is figure. Oh Lord, can I even fit it in the frame while I can read it? I don't think I can. Alright, feel free to pause the video, take a screenshot, and look at this information, should you want to. But that's just table 2, and that's just the results of the tests showing each model and the fixed effects tested. Oh, these are just stats. Oh, stats. Ew. Oh, the popping. I love popping. I really hope no one gets like grossed out like heavily by it. I love how it sounds. I want an ASMR video, just a chiropractor just like just someone's spine. Oh, I want that. Speaking of. Oh, only one. Cool. Great. So the results weren't that interesting to me. It just says if there is a virus in and around the area, it's getting to the others. They didn't really go into detail about how or why. It's just the virus is getting there. The discussion, the higher prevalence of both black queen cell virus and deformed wing virus in bumblebees near honeybee apiaries, the lack of finding of deformed wing virus in bumblebees at sites without honeybees, and the persistence of viruses on flowers collected only from sites with apiaries provides strong support for the hypothesis that RNA viruses are spilling over from managed honeybees into wild bumblebee populations through the shared use of floral resources. And I just want to go ahead and point that out. That was all one sentence. Scientists love their run-ons. Oh my fucking god. Alright, let's... Jesus Christ, this is... Oh, I'm just like almost done because I skipped the methods. Cool. I am going to finish this paper for you guys, and it's only like... Oh, we're only at 41, 42 minutes. It's cute. In addition, we detected replicating viruses in both Bombus bimaculatus and Bombus vaginus, demonstrating that bees were carrying active infections and not simply testing positive for virus particles they contacted, but might be cleared by passing through the gut. Okay, so they're getting these viruses from the gut. I'm going to turn my overhead light off so that way I don't have that weird looking like a halo. But you can't see. Oh, you can see my eyes just fine. Okay, cool. Thus, our study confirms that viral replication in 2 bumblebee species, adding to the list of bee species that may be affected by RNA viruses, may be affected. They fucking are. Oh my god. Alright, let me go ahead and show you figure 1. They really printed these out big boy style. Alright. So, let me open this. Oh, that was loud. I'm so sorry, guys. I'll, like, edit that down. Alright. When the apiary was present, which is the white bars. What? They're both apiary present. Oh, apiary absent in white bars. So there are no honeybees in and around the area. For black queen cell viruses in bumblebees, it's there. It's definitely there, but it's not anywhere near as high. When the apiary is present, it's a little higher. Same with the deformed wing virus, which is shown in this one closest to my hand. That's all that graph really wanted to show you. Sorry, I'm just going to massage your ears after that loud, obnoxious sound. Alright. Thus, our study confirms that viral replication... Oh yeah, I already read that. We detected bee viruses on flowers of 4 different plant species and only found viruses on flowers within honeybee apiaries. Okay, so the flowers are the means of how they're getting sick. These results support the hypothesis that viruses are left behind by foraging honeybees and provide evidence that sites near honeybee apiaries could be hotspots for disease transmission between honeybees and wild bees through the use of shared floral resources. If transmission of bee viruses occurs through the shared use of flowers, we predict virus prevalence patterns to be shaped by landscape-level floral composition. Oh, that's actually a good thing to point out. Okay, so I'm going to get closer and whisper this. So if the flowers are what's causing the honeybees to get, or the bumblebees to get sick from the honeybees, then just having honeybees and bumblebees near each other is not going to make each other sick. The flowers have to be there so we can track the virus through the flowers that the bees are visiting. Which is really important to be able to track where these viruses are going. Should I? Yeah. Give myself a little head massage. Oh, you can see a little bald spot forming. I love that for myself. Great. I just love a good tap. If transmission of bee viruses occurs through the shared use of flowers, we predict the virus prevalence pattern. Oh yeah. The prevalence of deformed wing virus in bumblebees was higher in sites with high floral density. Sites with high abundance of floral resources may become hotspots for transmission, as pollinators will converge to forage, and bee density or commingling may increase. Our results of deformed wing virus support this. Oh, there's another one. Phenomenon whereby the risk of infection was increased for individual foragers in areas of high floral abundance. However, we did not find an effect of floral density on black queen cell virus prevalence. Okay, so that means that black queen cell virus is probably transmitted differently, and they're gonna have to figure out how that's spread to track that one. So deformed wing virus found on the flowers. Black queen cell virus, I don't know yet. All right. Other factors besides transmission from honeybees at floral resources may be more important for the spread of black queen cell virus in bumblebees. Yeah. It is likely that black queen cell virus is vertically transmitted, as with honeybees, or highly transmittable among nestmates. Vertical transmission is transmitting between a species, or within a species. So that's like if I cough and give someone else the flu, that's vertical transmission. And especially in social colonies like bees, that's gonna be not ideal. In captive lab colonies that are positive for black queen cell virus, prevalence within a colony is near 100%, indicating that the rapid dissemination within a colony may occur. This may also explain our observation of high black queen cell virus prevalence as compared to deformed wing virus, as well as the occurrence of replicating black queen cell virus infections regardless of apiary presence. Okay, black queen cell virus is just way more infectious. In the bees we sampled, black queen cell virus prevalence and replication was higher in Bombus bimaculatus than Bombus vaginus. Although both species are medium-sized, long-tongued bees belonging to Pyrobombus subgenus, Bombus bimaculatus queens emerge earlier and establish colonies before Bombus vaginus. By emerging earlier, Bombus bimaculatus may have an increased opportunity for foraging overlap with honeybees and contracting virus particles on flowers. So that's kind of them trying to take a stab in the dark as to why one species was more infected than the other. They come out of hibernation earlier, so they have more time to interact with a virus. Not ideal, but it's not ideal, but things happen. In spring, honeybees intensify their pollen foraging activities to sustain brood rearing. Bees will forage for pollen so that they can feed it to their young, so that's where they get their protein content from. Nectar is mostly sugar, so it's used by adults for energy, whereas pollen is gathered, brought back to the nest and fed to larvae, so that way they can grow up and be big and strong. That's like their protein. Viruses are transmitted among bees through pollen. Early emerging bumblebees could be at higher risk for contracting contaminated pollen grains left behind by honeybees. Understanding the temporal variation of virus prevalence among bumblebee species and flowers would help to understand the ecological factors driving virus transmission and infectivity. Okay. Oh, I love how I look. I love this. It looks like a cat eye from right here. Oh. Several bumblebee species of Europe, North America, and Asia have suffered dramatic declines, particularly in North America. Pathogens appear to be a chief threat to this group. Overall, we detected deformed wing virus in 9.3% of all bumblebees tested, which falls between other estimates from Europe, which were reported between... Oh, I'm so sorry about that loud noise, guys. But the deformed wing virus is in between 3 and 11% in Europe, so we're in between that on the higher end, kind of, but great. However, black queen cell virus, which has a prevalence of 75%, was tested at 12.5 times higher than UK reports, which is only 6%. Oof. So black queen cell virus is going to really hit some of these native species of bees, and that's something that's got to be watched out for. Although it is often difficult to directly compare results among studies, we believe this substantial difference is notable given the similarities of sample size and sampling efforts between the studies. These differences could be due to bumblebee species susceptibility and or life history traits that affect exposure to viruses. That definitely could be. So what I got from that is, you know how when the Spaniards came over and gave the Native Americans smallpox, it really, 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 really hit the native populations because they had no resistance to it. Our bumblebees may have no resistance to this virus, and it could really hurt them. So that's definitely something to watch out for. These differences could be due to bumblebees. We hypothesized that in pooled flowers for virus assays. Separately testing petals, nectar, pollen, etc. could help understand where the virus is deposited on the flower and lead to experiment testing how different floral traits influence a plant species properly to harbor and transmit viruses. Oh my god. Oh, excuse me. For example, if viruses are detected in nectaries, antiviral secondary metabolites increased in the nectar of some plants could reduce virus viability. Okay, so they're saying that, like, once they figure out where it is in the flower, they can figure out, like, how to best help the bees by putting antivirals in the nectar. If it's the pollen, they can do something else. If it's the petal, they can, like, brush them off, stuff like that. Excuse me. Further, flowers with deep nectaries could exclude some pollinators and reduce transmission between bee species. Floral morphology that induces bee flower contact or forager handling time could also affect virus deposition. Future controlled experiments should educate how differences in floral traits influence the likelihood for virus deposition and transmission. Our results showed that both honeybee virus load and apiary presence are not important predictors of virus prevalence in bumblebees. These findings indicate a need for management guidelines that reduce the introduction and spread of bee pathogens. That is a true thing. Insects are really not protected by any sort of law whatsoever. We kind of threw bugs to the wind. We have protections for, and of course we do for, mammals, big animals, birds, fish. But bees are kind of we know there are so many of them that we kind of forgot how important they are. So that's why it's really important to care for the bees, to understand these guidelines, and to learn why these things are happening. These findings indicate a need for management guidelines, blah, 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 blah. Management guidelines could limit apiary activity or increase disease monitoring in critical habitats of selective wild bee populations. Although our study focused on 2 RNA virus, the spillover of numerous other pests and pathogens from commercial bees is well documented. Oh, that is well documented. 1, 2, 3, 4, 5 scientific articles already showing other variations of how this can be affected. Yeah, that's just something to put this in perspective. This is just 2 viruses they looked at. There is so much more. There is so much more out there than 2 viruses that can affect bees. Oh my god. It's and it's like this with humans. We got SARS, we got right now we got coronavirus, the variation of SARS. We got flu, we got E. coli, we've got, all sorts of things that can affect and hurt humans. And that's why understanding these things is so important. So that way we can prevent the infection, we can prevent the spread of it, and we can help cure those who are infected. I just really like the idea of helping people with knowledge, so that way we can prevent this, so that way people can live their happy lives doing whatever they want to do. That's just a little quick excerpt of why this is important. But with the interaction of global transportation of commercial pollinators, introduced pests and pathogens will continue to pose problems for conservation efforts, underlining the need to prevent the introduction of disease throughout robust monitoring and management practices. Sorry, I needed a hit. Full nicotine addiction, baby. That's what you get being a server if you want your breaks in North Carolina. Despite the burgeoning interest, interest of virus amongst wild bees, the effects of viruses on non-apis species physiology and fitness are virtually unknown. RNA viruses may contribute to the observed declines in bumblebees, or bumblebees may serve as tolerant reservoir hosts, facilitating the maintenance of viral infections within pollinator communities at large. In all, knowledge of the effects of RNA viruses and the conditions under which transmission among bee species occurs is a critical predictive understanding that informs the efforts to protect vulnerable species. Alright, so that's our paper, guys. Oh, and we made it in under an hour, and I did makeup. See, cutting out methods, kind of like that right now. But thank you guys so much for tuning into this GaySMR education about viruses and bees. I hope you guys have a wonderful night. I hope you learned something, or I hope you fell asleep and have joyous, wonderful dreams. I hope you guys have a wonderful night, and I will talk to you guys later. Bye-bye now.